So the way we make RNA visible in live cells is to use the very well established technique of introducing MS2 binding sites in a gene of interest, in a reporter gene. And what we have done was to introduce these MS2 binding sites in beta globin. We started by using beta globin as a reporter gene, and we took very care of just introducing one copy of this transgene in a cell line. So you will be looking at stable cell lines that have integrated a single copy of a transgene, and when the RNA is made, then it is bound by a red or a green fluorescent protein coupled to the MS2 protein. And when we look at these cells by spinning this confocal microscopy and we look at time lapses, what becomes obvious is that the intensity of fluorescence at the transcription site fluctuates over time. And because we have introduced the MS2 in an intron, these fluctuations reflect the synthesis of a new intron, and when the fluorescence comes down, the splicing of that intron. So in order to quantitatively analyze these fluctuations, we had to devise very precise methods for measuring the intensity of fluorescence at the transcription site. And as I mentioned, we use the spinning disk confocal, so we always record a stack of images around the transcription site, and we over time we measure what's the intensity at the best focal plane. In the type of plots that we get of fluctuations of fluorescence intensity over time look like this. This is how the raw fluorescence looks like in the microscope. These are the plots. And as I also mentioned to you, what we think is happening is every time there is an increase in fluorescence, this means that an intron has been synthesized and labeled by the MS2. And then after synthesis, the intron is spliced and degraded, and we have reasons to believe that this degradation of the intron occurs very fast and at the site of transcription. So when we see a decrease in fluorescence, this is the combination of splicing and degradation of the intron. But as you can see, we have several levels of fluorescence intensity, and because the same gene can be simultaneously transcribed by several polymerases, one problem that we had to figure out is how many molecules of RNAs are being simultaneously detected in these plots. So we use a trick to experimentally resolve how many RNAs we look at, and that trick was to treat cells with spliceostatin that has already been mentioned yesterday. It's a very potent splicing inhibitor, and we know that when we treat cells with spliceostatin, what happens is that unspliced transcripts are released from the site of transcription and diffuse around in the nucleus. So because now we can see single RNAs diffusing in the nucleus, we took advantage of this to measure what is the intensity of a single RNA. So this is now how it looks like, the little dots diffusing in the nucleus, and this is a still image from one of these time lapses, and this shows very clearly the difference when cells are not treated or treated with spliceostatin. And now what we have done was to quantify the fluorescence intensity in each of these little spots that are diffusing and that correspond to a single RNA. Then the next step was to convert the fluorescence intensity into number of mRNAs. And from that point on, we have just concentrated on fluctuations that correspond to about one transcript being made. And we selected in the plots from the time lapses, we selected those events, those fluctuations that start at 0, background fluorescence, meaning that there was no RNA at this point. Then there is an increase in fluorescence followed by a decrease back to background. So we assume this corresponds to the synthesis and splicing degradation of a single pre-messenger RNA. So at this point, we were confident that we could measure the lifetime of introns, and then we started to look at different types of introns. So first, because we were using the beta-globin gene as a reporter, what we have compared was the lifetime of the first and the second intron, because we can introduce MS2 bindings in either of the introns. And the first observation is that the total lifetime ranges between 20 seconds and 30 seconds for these two introns. I recall that these are quite short introns. But we can already detect a difference in time in kinetics between the two introns, and this was kind of expected and fit with a number of previous biochemical observations indicating that the removal of the second intron in beta-globin is coupled to 3' end formation. So it would be expected that it takes longer to splice the second intron. Also, we can introduce different types of stem loops. So we have introduced MS2 binding site and the lambda N binding site in the first and in the second intron. This allows to label the two introns with different colors, and when we simultaneously track the fluorescence in both channels, you can see that the signal co-localizes because we are always looking at the site of transcription. But what is very nice is you can see the first intron being synthesized and spliced first, and then the second intron. And this again also fits with previous biochemical data indicating that the second intron of beta-globin is spliced after the first intron. So from here, we moved on and we asked, can we now detect differences in the lifetime of introns that have splice sites with different strength? Because this has been a long standing idea in the field, is that if an intron has weak splice sites, it should take longer to be spliced. So here we moved to another reporter, and this reporter consists of two exons from the immunoglobulin gene, and there's been lots of studies done on the branch site sequence of these reporters. And what we have used, and I would like to thank Juan Valcárcel for giving us the backbone plasmids from which we did these constructions. What we use is we made two reporters, one with the endogenous, which is a weak polypyrimidine track, and we have replaced this polypyrimidine tract by a strong sequence taken from the adenovirals introns polypyrimidine tract. So we compared these two constructs. These two introns have exactly the same size and length. They just differ by the strength of the PI track. And now these are a couple of examples of how long it takes for them to be synthesized and spliced. And here you can see a number of examples. So this each of these plot corresponds to a different cell, and you can see that there is a clear difference in the behavior of the introns with a strong or with a weak splice site. The weak splice site, after it is synthesized, it takes a bit longer at the transcription site before the intron disappears. And this is the statistical analysis, and on average there is about a 10-second difference between the two. If all of this is correct, we should also predict that if you now increase the length of an intron, you should increase the lifetime, because now there are more nucleotides that need to be transcribed before the splicing. So in order to test this prediction, we moved on and we did another construct where now the splice sites are exactly the same, but we have introduced an extra 1,000 nucleotides after the MS2. So this means that when the polymerase synthesizes the MS2 binding sites, we start to see fluorescence, but then the polymerase has to synthesize an extra 1,000 nucleotides before the splice site is transcribed. And indeed, we do see a reflection of this time in the time it takes for the fluorescence to fluctuate. We do see an increased plateau of the fluorescence before it disappears when we look at these transcripts, and this is the statistics. So what makes these introns taking longer to be spliced? Is this just the rate of transcription elongation, or is it because it takes longer to splice? So we calculated how long does it take to transcribe the extra 1,000 nucleotides, and for this we had two options: either to use the transcriptional rates that have been reported in the literature from different type of approaches, or we also tried to measure transcription rate in our own system. I'm almost done. So in order to get a clue to transcription rate using our live cell imaging, what we did was to calculate how long does it take for the fluorescence to come from background level to maximum level. In calculating these time lapse, we come up with rates of transcription in the range of 4 kb, which fit many other studies that have been published. So, although the transcription rate may vary, if we assume 4 kb, which is a relatively consensual range of rate of obtain, then assuming a rate of 4 kb for transcriptional elongation, it would take 15 seconds to transcribe this extra 1,000 nucleotides. And if we look at the difference in the average time of the intron lifetime, it's almost in the range of 15 seconds. So although we have to dissect these differences with further care, we can say that clearly transcription, the transcriptional elongation is a rate-limiting step for the splicing of long introns, because the longer the intron is, the longer it will take to be transcribed. And we are also left with very few extra seconds, so less than 10 seconds for the splicing itself. So coming back to the question that Ian was asking yesterday, when we have a speed bump of polymerase, how long does it have to be stopped there in order to allow a weak intron to be spliced? I would say 10 seconds would be, an extra 10 seconds would be enough. Okay, so in summary, we have developed an assay that allows us to study splicing dynamics in living cells with single molecule, because we are just looking at a single transcript. We have shown that beta-globin introns are transcribed and spliced within a range of 20 to 30 seconds. We have shown that different splice site strengths do change the splicing kinetics, and that transcription is rate limiting for the splicing of long introns. And this work has been a collaboration between my lab, and we were introduced to the spinning disk live cell microscopy and single molecule analysis by Tom Kirkhausen, who very generously received me for a sabbatical in Boston. So I'm very proud to say that many of the time lapses that I presented to you were done by myself in the microscope. This study was possible thanks to a collaboration between the Portuguese Research Agency and Harvard Medical School. And in my lab, the people involved in making all the reagents and analyzing the data were Robert Martin, José Rino, and Célia Carvalho. And thank you very much for your attention.